We'll talk a little bit about the MHC, or major histocompatibility complex 1 and 2, and then we'll talk about the process of T cell activation. If you've been following this video series so far, again, everything is really going to start here in the bone marrow, and we're going to have, CD34 positive hematopoietic stem cells that eventually form the myeloid and the lymphoid lineage. And we're primarily concerned, especially in immunology, with the lymphoid lineage. And this lymphoid lineage is going to give rise to T cells, to B cells, to natural killer cells. Now, the B cells, remember, the B cells are going to not only originate in the bone marrow, but they'll also then go on to kind of develop in the bone marrow. But the T cells are a little bit different. They'll also originate in the bone marrow because they all come from the lymphoid progenitor cells, right, which are going to be found in the bone marrow. However, the T cells are a little bit different in that they'll develop in an outside lymphoid organ, and that is going to be the thymus. And you might be saying, well, how do you even get these lymphoid progenitor cells over to the thymus? Well, there's certain chemokines that are released that essentially are going to signal the lymphoid progenitor cells to shuffle over some of their inventory over to the thymus so that we can develop some T cells. Okay, so this lymphoid progenitor cell is going to leave the bone marrow through the bloodstream and eventually go to the thymus. Okay, so that's what we have here. It leaves the bone marrow, goes to the thymus. And at this point, this cell is CD4 negative, CD8 negative. There's no CD4, there's no CD8 on this cell. And these are two markers you want to be really familiar with a T cell, particularly in the stages of development, because different stages, there'll be different receptors, and this tends to be confusing. And the way I remember it is, look, this is a lymphoid progenitor cell. We haven't started kind of classifying it as a T cell until it gets to the thymus, right? And so at this point, we would expect it, as it arrives to the thymus, to be CD4 negative, CD8 negative. Now, once it gets to the thymus, though, there's going to be certain enzymes, certain genes that are going to stimulate shuffling of some of the genes in this precursor T cell. And the one that you really want to be familiar with is going to be RAG, which is essentially this recombinase activating gene. If you think about, a recombinase enzyme, what are we talking about? We're talking about taking the DNA, shuffling it up, mixing it up so that we get some variation and some diversity in those genes. And the idea here is, if we're shuffling up all of the DNA here in this precursor T cell, we can create a very unique T cell receptor that will bind to a unique antigen. If we create a whole bunch of different T cell receptors, we're going to have more immunity to different types of antigens. And that's essentially what these RAG genes are doing. And so the two big recombinase genes, like we said, is going to be RAG1 and RAG2 that are going to be kind of stimulating this process, this recombinase process, to eventually lead to the development of the T cell receptor. Now, remember what I said, you do have to keep in mind, where's CD8, where's CD4? And you can see here that this precursor T cell, after it comes into the thymus, it's going to have that recombinase gene causing the formation of a T cell receptor. And with that T cell receptor formation, there's also going to be the addition of CD4 and CD8. CD4 and CD8 are essentially co-receptors to the T cell receptor. Okay, so we're basically putting everything on this precursor T cell, including CD3. CD3 is usually going to be, in terms of board questions, a marker to tell you that you're dealing with a T cell. It doesn't necessarily tell you what type of T cell like CD4 and CD8 will, but CD3 is kind of just like a general T cell marker. So you're going to get a T cell receptor, CD4, CD8, and CD3 all placed on this precursor T cell once it gets to the thymus. It's essentially the body's way of saying, look, now that we've got this lymphoid progenitor cell here, we're marking this as a possible T cell. Okay, and so that's what all of this stuff going on to this T cell is for. Now, as you'll see in a minute, we'll eventually have a CD4 receptor or a CD8 receptor along with the T cell receptor and the CD3, but we won't have both CD4 and CD8 when this process is done. But in the beginning of the process, we have both of these receptors. And so sometimes these precursor cells, once they get to the thymus and they have RAG1 and RAG2 activation, they'll be called double positive T cells. Okay, and so just to recap this here, we have this initial pre-selection process, which I'll talk about in a minute. The T cell receptor will be created using RAG1 and RAG2, like we said, and it will be CD4 positive, CD8 positive. Now, the next step in this process is particularly high yield. This is positive selection. In positive selection, we're essentially trying to find the right antigen. You can think of it in some ways as a positive match in terms of an antigen that we can bind to that T cell receptor that we created using the RAG genes, RAG1 and RAG2. And so just to take this for example, what will happen is, again, the blue structure here is going to be your T cell receptor. And you can see that T cell receptor can bind to some of these thymic epithelial cells. Okay, so this would be a thymic epithelial cell. And these thymic epithelial cells will either have MHC1 or MHC2. Okay, now remember, the CD4 positive co-receptor will help that T cell receptor bind to MHC2. Conversely, the CD8 positive coreceptor will help that T cell receptor bind to MHC1. Okay, and again, this goes back to your rule of 8s. Okay, so 8 times 1 is 8, right? So we're talking about CD8 positive times MHC1. That gets you to 8. Now on the other side of that, 4 times 2 also equals 8, right? So CD4 positive cells that are going to bind to MHC2 receptors will also get you to 8. Okay, so that's the way to remember. CD8 positive cells are going to bind MHC1. CD4 positive cells will bind MHC2. And you might be saying, okay, hold on a second there, Dr. Visor. You said that this cell has CD8 and CD4. So how do I know which one is going to bind? In reality, one of these receptors will find their specific antigen on these thymic epithelial cells and they'll bind. And when they do that, they're going to essentially select themselves to either be a CD4 or CD8 positive T cell. Okay, so it's kind of, in some ways, it's a bit random how this happens. But the point is, the big point that you want to remember here is that in the process of positive selection, what we're essentially doing is we're finding the right fit. We're finding the antigen that binds specifically to this receptor. Now, if that antigen binds too tightly or it doesn't bind strong enough, what will happen is these T cells will eventually get an apoptosis signal. So if the binding is too strong or too weak, that T cell will eventually undergo apoptosis. You have to have just the right amount of binding to get to the next step, which would be negative selection. Okay, so in positive selection, you're looking for adequate binding. And again, the CD8 positive coreceptor will bind with MHC1, the CD4 positive coreceptor will bind with MHC2. So it's kind of completely random as to if, this precursor T cell binds to CD4 positive MHC2 or uses CD8 positive to bind to MHC1. But once that process happens, then we're essentially selecting the route that this cell will go down, okay, once it chooses to bind to whatever specific antigen is present. And again, if the binding is too strong or too weak, then it's going to undergo apoptosis. But if the binding is just right, then we can get to the next step. And the next step is going to be negative selection. Now, negative selection, unlike positive selection, is going to take place in the thymic medulla. Again, we're going deeper into the thymus as we proceed here. Okay, so negative selection is the process of essentially, again, binding to the same type of receptors that we bound to in the initial step, but the difference in negative selection is that we're using our own self-antigen. So these medullary thymic cells, instead of presenting a foreign antigen, we're presenting antigens from our own body. And the idea in negative selection is we don't want our T cells to attack our body, right? We don't want our T cells to attack our own cells. And so we're presenting our own cells, kind of like a test to say, hey, we just want to make sure that you're not going to be attacking us. Okay, and so that's what this second step is. Now, if the T cells bind really tightly to our own antigens, that's concerning because then we're thinking, well, it's possible that these T cells could attack our own cells. Okay, so self-antigens, again, self-antigens being the key point, right? Antigens from our own cells presented on the MHC1 and MHC2 receptors. If strong binding occurs to self-peptides, again, our own peptides, then those T cells will likely undergo apoptosis. So you can see that again here. So if the binding is really strong, again, these T cells will then undergo apoptosis, okay? Now, the T cells that do not bind, right? So the T cells that do not bind, those cells will then go on to form mature cells, okay? So again, this being the example of the CD4 positive T cell, and then this being the example of the CD8 positive T cell. And again, in both situations, if there's no binding, then we go on to essentially have a matured T cell, okay? Once we complete negative selection, we have a matured T cell. Now, keep in mind, this T cell is still naive. What does that mean? Naive means that this T cell has not yet run into its antigen that binds to its specific T cell receptor in the body yet. Okay, once this T cell runs into that antigen, that T cell can then become activated. Okay, typically when a macrophage or a dendritic cell presents that antigen to the T cell and activates it, then that T cell would no longer be naive. But at this point, it's important to understand the terminology. The T cell is mature because it went through this whole process. It's been tested for positive selection. It binds to its antigen, not too strong, not too weak, and it won't attack our own cells, most likely because we didn't have binding to self-peptides. Okay, so that's the concept. So that's why it's mature. It's naive because it has not yet run into its antigen in the bloodstream. So just keep that in mind, or in any lymphoid organ in the body. What you'll notice here at the very end of this, you might be saying, what happened to those CD4, CD8 receptors? Okay, so you can see here, we had a CD4 positive cell in negative selection. It still had on this CD8 receptor. So basically, throughout this whole process, these cells were double positive T cells. Through positive selection, through negative selection, they were double positive. But what will happen is, after the selection process has occurred, you'll start to see downregulation of whatever receptor was not used during this process. So if we look on the left, this was mostly going to be the CD4 positive T cell because it bound to MHC2 initially, right? Going back to our rule of eights. So what happened is, eventually you'll see that this CD8 co-receptor eventually falls off as it matures into a mature T cell. Okay, so when you get to a mature T cell, you don't have CD4 positive, CD8 positive. You have one or the other. It just depends on which pathway this T cell went down. So you can see here, once you have a naive T cell post-selection, an immunocompetent mature T cell, it's either going to be CD4 positive, CD8 negative, or CD4 negative, CD8 positive. Again, if it's CD8 positive, we call those cytotoxic T cells, CD4 positive T helper cells, and we'll go into a lot of detail on this in the next couple videos. But the point is that there's downregulation of that receptor after we go through selection. And the last thing that I want to touch on here, remember we talked a little bit about these Tregs or T regulatory cells. Now, a small percentage of these cells that actually do bind to self-antigens will actually stick around. So you might be saying, what are we talking about here? We're going back to this step at negative selection. Okay, so right here, again, we presented our own antigens, a self-antigen. We don't want the T cell receptor to bind, right? When the T cell receptor binds to that, we basically are saying, hey, this thing has got to go. It's got to undergo apoptosis because it could attack us. A small percentage that actually do bind don't undergo apoptosis and instead go on to form regulatory T cells. Okay, and this is particularly classic for the CD4 positive line here on the left. So what these T regulatory cells essentially do is they help kind of damper down the immune response. They regulate the immune response, basically so it just doesn't get out of control. Okay, that's kind of a simple way of explaining it. And again, we'll go into more detail on this coming up. But the important thing to know here when we're talking about this self-antigen presentation with regulatory T cells and negative selection is there's a very classic protein here, and that's going to be this ARI protein. And this is very classic because this particular protein is responsible for allowing this process to happen. In other words, it allows us to give self-antigen presentation so that we can kind of show these T cells, hey, this is what our antigens look like. If this protein is not functioning accordingly, if there's a defect in this, then you can imagine we might have a higher risk of essentially attacking our own cells because we're not able to effectively undergo negative selection. And the classic pathology that this manifests as is going to be autoimmune polyendocrine syndrome, which is essentially a combination of a bunch of different endocrine-like disorders due to this autoimmune response where we're attacking a lot of our own cells, leading to endocrine dysfunction. So you get things like adrenal insufficiency, you can have hypoparathyroidism, chronic mucocandidiasis is also very classic. And again, we'll probably talk about that in greater detail, but just kind of keep that in mind, big picture. Okay, so just to recap this, right, we had some lymphoid progenitor cells, we had chemokines that brought that lymphoid progenitor cell from the bone marrow where it originated to the thymus to mature. Once it got to the thymus, CD4 negative, CD8 negative, the recombinase gene shuffled up the DNA, we made a T cell receptor, and it was double positive at that point. And again, I want to say that all of these, if you look at all of these, they all have this CD3 receptor on them. So throughout this entire process, right, we also had a CD3 receptor, and that stays on all of these cells even when they're mature. Remember, CD3 is very classic for marking a cell as a T cell in general. Okay, so remember that. But the CD4 and the CD8 receptors, we're going to basically go down one of the two paths where we end up keeping one or the other. Okay, so again, if we initially are going to bind to MHC2, thymic cortical epithelial cells, we'll then probably go down the CD4 positive route. If we bind to MHC1, we'll go down the CD8 positive route. You can always remember that with your rule of eights. Okay, so the first step was positive selection. We wanted to see if there's adequate binding to an antigen that is not self, right? Some other foreign antigen usually. And if there was adequate binding, the T cell will go to the next step. If not, it would undergo apoptosis. With adequate binding, the T cell moves to the medulla. Then there's negative selection. We present self-antigens largely driven by this protein, the ARI protein. And if there's binding to self-antigens, then that cell needs to undergo apoptosis because we don't want it attacking self. Some of the small percentage of those cells will become Treg cells to help regulate the immune response. And then eventually, once this process is completed and this cell survives, then it will become a mature T cell, either CD8 positive, CD4 positive, and then downregulate the other co-receptor. It will still have a T cell receptor and it will still be CD3 positive. And so what is this major histocompatibility complex anyway? It's really just another form of communication. It's a communication receptor. We talked about cytokines kind of assisting with communication, and the MHC complex is going to be very classically associated with binding to these T cell receptors, and usually the MHC receptor is either going to be on kind of like your everyday nucleated cell or an antigen presenting cell. Now, when we're talking about class 1 MHC, we're talking about the kind of the standard MHC receptor. That's how I remember it. Class 1, it's kind of just like your baseline MHC receptor. It's pretty much going to be on almost all of your cells, particularly most nucleated cells. So remember, when we say nucleated cells, we're not talking about red blood cells, right? Red blood cells will not have MHC1 because they're not nucleated. Okay, so this has repercussions down the road when we talk about some of the pathology, but that's just something to keep in mind. Now, the key is the MHC1 receptor is essentially looking at every cell, and it's making sure that there's kind of nothing suspicious going on with all of our cells. So it allows for recognition of healthy versus infected cells. So you can imagine, right? So if you just have some normal cell here, okay, so this normal cell is doing what it's supposed to be doing, and on its surface here it has this kind of MHC1 receptor, and if I have some T cell nearby, okay, so I have some T cell over here, this T cell will also have a T cell receptor, and this kind of healthy cell might present some of the self-antigens. So there's a lot of proteins and that type of thing that are in the cytosol of the cell, and some of those proteins might end up going into the rough endoplasmic reticulum and eventually getting transported to the surface of this MHC1 receptor. And this T cell might recognize that and say, hey, okay, that's a self-antigen. And remember, we already talked about this. In negative selection, we had our T cells undergo negative selection, right, in the thymus when they were maturing. And in negative selection, we tried to bind to self-antigens, but the T cells that survived did not bind to self-antigens. So this T cell that's then seeing this healthy cell putting a self-antigen here, it's not really going to bind to it that tightly. And so there's nothing to worry about, right? Now, let's see what happens, though, if I had a virus come around. So if I had some virus come around, okay, so here's some virus. All right, and this virus then gets into this cell. So it's going to go into the cell, it's going to hijack the cell's machinery so it can make more viral proteins. Well, the cell, what it will do is it will take some of these proteins, like we just talked about, it'll bring them into the rough endoplasmic reticulum, okay, and then it'll break them down and then transport them to the surface of the cell. And so now when this T cell is kind of jumping around here, checking out all the cells, it's going to see, hey, look, there's an antigen that's presented here that isn't really supposed to be here. It's going to bind to this antigen because it's a foreign antigen. It's going to be the right antigen to match that specific T cell receptor. And then we'll talk about what this T cell does, but you have to keep this in mind, right? On all of these kind of normal everyday cells, they're going to have MHC1 receptors, right? They're going to have MHC1 receptors. So that means that this cell that's binding to MHC1, what did we say? Well, it must be CD8 positive according to the rule of 8s, which means that this must be a cytotoxic T cell, cytotoxic killer T cell. Okay, and so you'll see what we'll talk about in the next video. This cytotoxic T cell will then essentially destroy this cell using granzyme and using enzymes to essentially punch big holes into the cell membrane, because the idea is this cell, we're basically sacrificing it because we don't want this virus to then kind of burst that cell and then have even more viral particles infecting other cells. So that's the concept. And remember, what this cell will also do, it will also release some interferon alpha and interferon beta to the nearby cells. Okay, and so it'll essentially try to tell them to upregulate their MHC using interferon alpha and interferon beta. Remember, this will then upregulate MHC1 on the nearby cells so that there's a higher probability that if those other cells are infected, they will be detected early by more cytotoxic T cells. MHC is not just in humans, okay? So the way that we kind of classify it or grade these, we call these the human leukocyte antigens. And it's just important for you to understand that for MHC class 1, you can see there's only one letter after this human leukocyte antigen or HLA class. So the specific one here, A, B, C, there's only one letter, okay? So one letter for MHC class 1. You'll see in MHC class 2, there's 2 letters. So that's how you remember it. One letter, think MHC class 1, 2 letters, think class 2. Now sometimes you'll get questions on the actual structure of the MHC receptor. We can make a whole 2-hour video on, the different receptors, but just make sure you understand that these receptors have beta 2 microglobulin. Okay, so that's this component here. And you can see that the MHC1 receptor is kind of asymmetric compared to the MHC2 receptor. The MHC2 receptor looks a little bit neater. It looks a little bit more symmetric. So sometimes you'll see that, this MHC1 receptor, they'll say it's either asymmetric or has 2 polypeptides of different lengths. So just kind of keep that in mind. There's a little bit of asymmetry here, and it also uses beta 2 microglobulin. Now, the MHC1 receptor complex, like I was talking about here, it's developed in the rough endoplasmic reticulum, which is a little bit different than the MHC class 2. So again, these are just kind of some of the details that sometimes come up that you want to be familiar with. And the key thing that we talked about here is that, again, a virus has to usually infect a cell, right? So it's usually an intracellular pathogen, something that infects the cell so that then we can, once it's in the cell, we can take its proteins, put it in the rough endoplasmic reticulum, eventually get it through the Golgi back up to the MHC1 transporter. So again, this is going to be, MHC1 is going to be usually intracellular pathogens for that reason. Okay, and again, CD8 positive T cells are the ones that are going to recognize MHC1. We talked about that already, usually leading to cell-mediated destruction of that cell. Now, the particularly high yield thing, this is very high yield. If there's an absence of an MHC1 receptor, in other words, if the T cell comes by and says, hey, this guy doesn't have an MHC1 receptor, if there's a cell that doesn't have MHC1, natural killer cells will tend to destroy those cells. Okay, so natural killer cells are kind of on the hunt to make sure everybody's got their MHC1 receptor. And you might be saying, well, what's the point of that? Well, remember, if you have a virus that comes by and infects a cell, through evolutionary processes, these viruses have learned to downregulate MHC1 receptors, right? Again, we're releasing interferon alpha and beta because we want more MHC1 receptors, but the viruses have trying to downregulate them. Everybody's trying to kind of compete to see if these cells have an infection, okay? And the viruses obviously don't want to be detected, okay? So they can downregulate MHC1. The same thing is true for cancer cells. Cancer cells can also do this. Natural killer cells are always on the look for an absence of MHC1. And again, MHC1 receptors will be upregulated by interferon alpha and beta, and I think we talked about that earlier. Now, when we're talking about MHC class 2, we're not talking about just your kind of like everyday nucleated cell. We're talking specifically about antigen-presenting cells. Remember, we said this is going to be macrophages, dendritic cells, B cells, right? These are all going to be very classic for presenting antigens to either B cells or T cells. And again, the HLAs are going to be 2-letter HLAs, specifically for MHC class 2. Now, again, we talked about the receptor has chains of equal length, which is going to be different from the receptor in MHC class 1, which is going to be asymmetric and have beta 2 microglobulin. The other thing that sometimes comes up here when we're talking about MHC class 2 is this invariant chain. When we talked about MHC class 1, we're talking about an intracellular pathogen. For MHC class 2, because we're talking about antigen presenting cells like this macrophage here, this macrophage, right, how is this process going to start? This macrophage is eventually going to get into some contact with some bacteria here. Okay, so let's say that this is a bacteria. Okay, so this macrophage is going to get into contact with this bacteria. It might attempt to destroy it or phagocytose it. And once it does that, it's going to break that bacteria up in the phagosome, right? And eventually it'll fuse with the lysosome to break down that intracellular bacteria. Okay, so it's going to kind of keep them in these phagolysosomes to break down the bacterial contents. So that's what the macrophage is initially going to do. And we already kind of talked about how this process originates. Now, once you get this bacteria in this phagolysosome, you're going to acidify it, right? Macrophages, we also talked a little bit about the superoxide radical formation, respiratory burst pathway. So you're essentially breaking this bacteria down. Now, once you break it down, this macrophage is going to take components of that bacteria and present it on MHC2. Okay, and so once you do that, now you can have a CD4 positive T cell come around, bind to that MHC2 receptor, detect it, right? And then it can activate this macrophage. And it does that through interferon gamma classically. Now, notice what's different here. When we talked about the first pathway, we talked about this whole thing happening because it was an intracellular pathogen that infected the cell, that being the virus. Here, it was an extracellular pathogen that a macrophage digested and then presented to MHC2. Now, in the process of this happening, there's kind of like a little chain on the MHC2 receptor inside of this phagolysosome, and that's this invariant chain. And the invariant chain just essentially prevents the antigen from binding prematurely. And it's one of those things that, I wouldn't remember every step of the process unless you have a lot of extra room in your brain, but the invariant chain is one component that I would associate with MHC class 2. So think of it like a little hook on this MHC receptor as it's in this phagolysosome before it gets transported to the cell surface that essentially prevents the antigen from binding before it's broken down completely. Okay, so that's just a simple kind of way to remember that. And remember, when we're talking about MHC class 2, extracellular pathogens, CD4 positive T cells, usually resulting in a humoral response, at least primarily a humoral response. Whereas when we're talking about MHC class 1, we're talking about intracellular pathogens like viruses in particular that result in CD8 positive T cell mediated destruction, usually more of a cell mediated process. The goal here is to essentially take the concepts that we've been talking about and then integrate them into this slide. Okay, so let's start off with the basics here. So cytotoxic T cell activation, which we kind of just talked about, very good versus intracellular pathogens. Again, we have our kind of everyday nucleated cell here. It's usually going to be presenting self-antigens on MHC1. However, if this cell was infected with some kind of virus, for example, right, like we just talked about, if there was some virus around that infected the cell, okay, here's my virus infecting the cell, then that viral antigens might be presented on MHC1, CD8 positive T cells, again, binding to MHC1. These are going to be cytotoxic T cells. They'll recognize that the cell is infected by some intracellular pathogen, and they'll release perforin and granzyme B to destroy the cell. So perforin, just think of perforating the cell membrane, right? So these are essentially going to be molecules that are going to be cutting holes into the cell to essentially destroy it, to prevent this virus from hijacking more of the cell machinery and eventually leading to a terminal lytic cycle in the cell and then kind of reproducing accordingly. And so that's what the cytotoxic CD8 positive T cell is doing. It's trying to destroy the cell because it's infected, again, through this process that we just talked about. So I think we've hit on that a couple times. Now, the important thing to remember is that, again, the antigen that's presented was endogenous. In other words, it came from inside of the cell. It was a viral protein. Usually we expect self-antigens to be presented in MHC class 1, and normally our body should not be binding to those because we went through negative selection. However, think of one of the correlations of this, and this is kind of some next level stuff, but if you have a patient that has a transplant patient, for example, or you put a donor graft in a patient, so let's just say you put a graft in a patient, and now that patient has somebody else's cells, right? And if that person doesn't have, the same type of HLA receptors there, right, they're going to present different self-antigens. And so what's going to happen is if you put a graft in another patient and they have different MHC1 receptors or different variants of these, right, different HLA variants, and the person that received the graft, right, that person has T cells, right? They have CD8 positive T cells that could attack the grafted cells. So you can imagine, let's say that this cell, right, this cell came from the donor. Okay, and then this CD8 positive T cell, let's say that this was from the host. Okay, so this is the host or the recipient that received the graft. So what can happen is, again, this MHC receptor might be somewhat foreign, right? It might not have the same exact HLA compatibility. And so these host cells will recognize, hey, this is not a self-antigen, and they'll actually start to destroy these donor cells for that reason. Okay, and so this is something that you'll sometimes see. And then again, there's variants of this. We'll talk about all of this in transplant rejection, but essentially this is what's happening. If you don't have self-antigens on MHC1, those cells could get attacked. And that's why we immunosuppress patients after they get a kidney transplant or bone marrow transplant, because you don't want the immune system attacking these cells. But then that does open the door for a virus to potentially start to infect these cells without any regulation if you're immunosuppressed. But I'm going to get off the soapbox there and we'll go back to the next topic here, which is going to be the helper T cell activation. Now, one thing that's going to be a little tricky here, when you're talking about helper T cells, you can go two different routes. You can go a lot of different routes, actually. You can go T helper 1, T helper 2. Those are the big ones for step 1. But then, of course, there's regulatory T cells, there's T helper 17 cells, etc. But what I just told you earlier, okay, and again, we're just kind of adding layers to this, but what I just told you earlier is usually when you're talking about T helper cells, number one, this is CD4 positive, meaning that there's going to be MHC2 binding to CD4 positive T cell receptors, and this is usually going to be from an antigen presenting cell, right? An antigen presenting cell binding to a CD4 positive T cell, okay? So that's kind of what we've already talked about, and you can see it here. Here's the MHC2 receptor, here's the CD4 positive co-receptor, here's the T cell receptor, but there's also going to be a B7 CD28 binding. Okay, and so essentially this is going to be a co-stimulatory signal. It's essentially going to help activate this T cell. Okay, and without that co-stimulatory signal, there's going to be a significant drop-off in the ability for these T cells to get activated. And so this is just one of those things that you want to be familiar with. Now, B7 can also be referred to as CD80 or CD86. Okay, so just kind of keep that in mind. And a lot of people remember this as saying, look, 7 times 4 is 28, right? 7 is B7, 28 is what it's binding to, and the 4 is a CD4 positive T cell. Okay, so again, these are all kind of ways to, just remember this, because a lot of these are kind of confusing, and it might help to use some flashcards, go through some of the recall questions on here. But again, that's the general concept. So antigen presenting cell presents an antigen via MHC2, like we talked about, to a CD4 positive T cell, and there's a co-stimulatory signal that's B7 CD28, right? 7 times 4 is 28. Now, the thing that I told you earlier, I was kind of only giving you the half truth here. In general, when you stimulate helper T cells, there's usually going to be a very effective response versus extracellular pathogens, right? We talked about this. If it's an antigen presenting cell, usually like a macrophage, right? They're usually going to run into things like bacteria, okay? And they'll digest this bacteria, right? They're going to eat up this bacteria, and then they're going to put the bacteria in these phagolysosomes that will essentially get acidified. Once the invariant chain kind of pops off, they'll present these antigens on their MHC2 receptors to T cells. Okay, and again, these were extracellular pathogens that they're presenting, right? So this was an extracellular pathogen. And usually what will happen is in these situations, these antigen presenting cells will then release interleukin 4, like we talked about earlier. And the interleukin 4 will stimulate this T cell to become a T helper 2 cell. Okay, so again, this is going to be stimulated by interleukin 4 released from the antigen presenting cell. And then these T helper 2 cells, right, once this becomes a T helper 2 cell, that T helper 2 cell can then go on to release more interleukin 4, it can release interleukin 5, it can release interleukin 13. Okay, and we already kind of talked about some of the implications of this. These are all going to help stimulate eosinophils, basophils, mast cells. They're going to help cause class switching on B cells, which we'll talk about in the next video. Okay, so that's kind of the whole process. So when I think about helper T cells, this is really the process that I'm referring to when I'm thinking about helper T cells. Now, but we can't forget that there are also T helper 1 cells. You might be saying, well, how is that different from the T helper 2 response? Well, the T helper 1 response is pretty interesting. The T helper 1 response, let's just erase some of this here. So in the T helper 1 response, remember the difference is that we're going to be using interleukin 12 to stimulate this naive T cell to form a T helper 1 cell. Why would this antigen presenting cell want to release interleukin 12 instead of interleukin 4? Like, what would cause that? Well, let's just say, for example, that I'm dealing with an intracellular bacteria. Okay, so something a little different. Let's just say that I have an intracellular bacteria here. Okay, so not talking about a virus. What intracellular bacteria is very classic that we talk about very frequently? How about Mycobacteria tuberculosis? Okay, I'm just going to put MTB here for tuberculosis. Okay, remember, this is acid-fast intracellular bacteria, and a lot of times our body doesn't know how to destroy tuberculosis, right? And so this is going to be an intracellular pathogen. Okay, so it's intracellular in the antigen presenting cell, not necessarily affecting all of the cells systemically, but more classic for antigen presenting cells. But again, it's intracellular, right? The body digested this bacteria, but it doesn't really know what to do with it. It's having a hard time breaking it down. And so it's just going to kind of hold it here and present some of those antigens. And in the process of doing that, it's going to say, hey, we're dealing with something different here. It might release interleukin 12 instead to this T cell, causing this T cell to become a T helper 1 cell instead of a T helper 2 cell. Now, once we get a T helper 1 cell here, it might stimulate this macrophage and say, here, let me give you some extra juice. It'll give it some interferon gamma to activate this macrophage. Okay, and again, these macrophages can eventually go on to form epithelioid macrophages, giant cells, and the whole idea is we're essentially going to use these macrophages to kind of help digest and wall off this infection. And that's where you get your granuloma formation, okay, which is very classic for this T helper 1 response. And the key is that this all kind of happened because, again, this was an intracellular pathogens. So in the last video, when I talked about MHC complexes, I said in general, if you're talking about CD4 positive cells, you're usually talking about extracellular pathogens that you're digesting. However, with the T helper 1 response, this is more so specific for intracellular pathogens. So I know that's kind of confusing, but just keep that in mind. And usually this is going to come up when we talk about things like granulomas or viruses that are potentially infecting antigen-presenting cells. And these T cells will go on, like we said, to release interferon gamma to the macrophage to activate the macrophage, and we'll also get some interleukin release and then TNF-alpha. Now remember, the other thing I want to say here is interleukin 2, when we talked about cytokines, you're going to get some interleukin 2 release pretty much with almost all of these different steps. Interleukin 2 is going to stimulate all immune cells, T cells, B cells, natural killer cells, etc. And the key thing to remember the high yield thing about interleukin 2 is it can also have autocrine action. It can actually kind of stimulate the same cell that released it. Okay, so we talked about that in the cytokine video. Finally, the last thing on here is going to be these regulatory T cells. And regulatory T cells, like I said, they're kind of going to help damper down the immune response. These are essentially going to come from CD4 positive T cells that had some binding to self-antigens. Okay, and so these regulatory T cells are very cognizant of that. So they're protecting us from self-antigens, preventing autoimmunity. That's the key with regulatory T cells. So you can imagine if you had some defect in your regulatory T cells or in the process of negative selection, you might end up with an autoimmune phenomena, right? So you can see the CD4 positivity in these cells, which kind of tells you where they came from. But the key to look for if you're trying to distinguish these cells from others, CD25 positive, FOXP3 positive are very classic for regulatory T cells. And again, if we're bringing down the immune response, right, we're going to be releasing things like TGF-beta. Okay, this is kind of like the opposite to the interleukin 2s of the world, the interleukin 1, the interleukin 6, right? This is going to be the complete opposite response. Don't get TGF-beta confused with TNF-alpha, right? TNF-alpha, we're stimulating, upregulating immune response. TGF-beta, downregulating immune response. And again, these regulatory T cells will be inhibited by interleukin 6. That should make sense. Interleukin 6, pro-inflammatory. TGF-beta is going to be anti-inflammatory. Now, once these cells are stimulated, they can release more TGF-beta, right, to increase the anti-inflammatory response, but they'll also release interleukin 10. So the big two to remember for anti-inflammatory response is going to be TGF-beta, interleukin 10 to decrease the immune response. Kind of a lot of information here, but I think this is really pretty much all you need to really nail down T cells, even though it's such a big topic. And in the next series, we'll take this one step further with B cells to see how we end up developing antibodies. Take 15 seconds to pause the video, read the question, and then we will go through the answer. All right, so we have a 10-month-old female who's presenting to her pediatrician with a 4-day history of irritability and decreased appetite. Okay, her mother reports that her symptoms were accompanied by a fever that resolved yesterday. Shortly after the fever subsided, a generalized erythematous rash on the trunk was noted by the family. Shortly after this fever subsided, now the patient has a rash that is new, that's proximal, right, starting here on the trunk. Since that time, the rash has spread to the face, neck, and lower extremities, so spreading distally from the trunk. Immunizations are current, okay, that's kind of an important point, and the family denies any recent illnesses or sick contacts. On examination, the infant is well appearing with stable vital signs and a blanching maculopapular rash extending from the trunk to the bilateral lower extremities. CD56 positive lymphocytes in this patient most likely targeted infected cells through which process. Okay, so there's a couple things going on here. First of all, like a lot of these questions, and again, and this is kind of the evolution of step one questions. You don't necessarily need to know everything that's going on in the question to get to the right answer, right? If you kind of understand, first of all, what is a CD56 positive cell? When you see CD56 positive, right, the first thing that should kind of jump in your mind when you see CD56 positive, you want to immediately start to think about natural killer cells, okay? Natural killer T lymphocytes specifically. So these cells, they have a couple different functions, but one of the major functions of natural killer cells that we've kind of touched on so far is that natural killer cells can kind of go around and sample host cells and look at their MHC1 receptors. Okay, so for example, our regular cells, right? So maybe these are just some cells not necessarily directly involved with the immune system, but these cells typically, right? All of the nucleated host cells should have an MHC1 receptor, right? So these cells should have an MHC1 receptor. Now, typically what will happen is if the host, for whatever reason, is infected with a virus, right? So we'll just say there's some virus here, right? So this is my virus, and this virus, let's just say this virus hijacks this cell's machinery, right? So now this cell is actually producing viral particles for this virus, right? And so eventually what happens is the host will then digest some of these particles, bring them up through the rough endoplasmic reticulum, and transport this eventually to the MHC1 receptor, right? So these viral particles will actually get placed on the MHC1 receptor. Now, this is a risk for the virus because once these particles get presented on the MHC1 receptor, now this cell is basically known to harbor this virus. So if a CD8 positive T cell came around, which remember, these are these cytotoxic killer T cells. Remember your rule of 8s, right? So MHC1 is going to bind to the CD8 T cell receptor, right? 1 times 8 is 8, right? So this CD8 positive receptor, right, with a similar idiotype can bind to that MHC1 receptor and then realize that, hey, this cell is infected with a virus, and then start to release granzymes, perforins, right, to destroy this cell. Okay, so that's the concept. What this virus can do to try to counteract this whole process after infecting a cell, so let's just use a different color here. Let's say that we have a smarter virus, so to speak, on this side. So this virus infects this cell. The difference is, right, once this virus starts to kind of have the cell produce its viral particles, what this virus can do is it can actually code to downregulate the MHC1 receptor. Okay, so it can code to downregulate this MHC1 receptor. So now the CD8 positive T cell that's kind of coming around here, it's sampling the different MHC1 receptors. Well, guess what? There's no MHC1 receptor on this host cell. And so it's not able to detect the viral particle there, for example. Now, what the natural killer cells can do is they can come in. Remember, these are going to be these CD56 positive cells, right? These cells can come in and they can identify that, hey, this cell does not have an MHC1 receptor. Okay, and that's particularly problematic. And actually, the natural killer cells have various mechanisms also of destroying host cells that are not displaying MHC1, which is significant not only for viruses, but when you're talking about things like cancer, for example, right? So cancer can do something similar. Cancer can downregulate MHC1 receptors so that it can go undetected. When you see questions where they're referencing viruses in general and CD56 positive cells, the thing that you want to think about is downregulation of the MHC1 receptor on tissue, right? How is the CD56 positive lymphocytes targeting the infected cells? It's targeting the infected cells by identifying the lack of MHC1 receptors. And then, you could take this a step further if you really want to kind of understand what's going on here, right? So you have a 10-month-old, okay, 10-month-old who's coming in. She had 4 days of kind of irritability, decreased appetite, kind of non-specific symptoms. We might be suspecting something viral potentially. She had a fever, but to really kind of nail this down, the key line here to figure out what's going on, shortly after the fever subsided, a generalized erythematous rash on the trunk was noted by the family. That is very, very suggestive, especially in this age range, of HHV6 usually, sometimes HHV7. And this is an example of roseola infantum. So the key point here, the key takeaway, number one, you're looking for, kind of non-specific symptoms with a fever for like 3 to 5 days, somewhere in there. And then once this fever breaks, right, once that fever breaks, now the patient presents with the rash, right, as soon as that fever breaks. Okay, the second thing that you should remember too when you're talking about HHV6 or HHV7, a lot of these different viruses, you kind of want to understand where the rash starts and where it ends, and then, a little bit about the rash. So in this case, this is going to be a proximal, usually in the trunk is where this will start, or maybe the thighs, and then it's going to spread distally, right? The rash is going to spread distally, very similar to what we see here. So this is very classic. But again, the giveaway line here is definitely the fever breaking. Now the patient has an erythematous rash. That's blanching. That's maculopapular in nature. This is all very classic for HHV6. Now there's other, viral syndromes that potentially could show up in a question like this. You could think of something like, for example, measles, where you might have coryza, cough, Koplik spots. You can also think about something like erythema infectiosum from parvovirus B19. But that typically would be present in an older child, usually like 9 to 12 years old. When you're talking about a 10-month-old, HHV6, HHV7, definitely in that like 9 to 21-month age range is very classic, especially like we said with this classic presentation. Now, let's just quickly walk through the other options here. So we already said absence of MHC1 receptor is the correct answer. Interleukin-5 release. Remember the big thing, about interleukin-5 that we've kind of touched on is that interleukin-5 actually assists with class switching, right? When we have our CD40 ligand binding, the interleukin-5 cytokine can kind of stimulate the B cell to become a plasma factory that then produces IgA antibodies. Interleukin-4 release, on the other hand, is kind of going to do the same thing, but it's going to more so promote IgG and IgE class switching antibodies. Now again, class switching isn't really classic when we think about natural killer cells, right? We're talking about CD56 positive cells. If we said something about CD4 positive T helper cells or something along those lines, that would be more classic for a class switching type mechanism. But again, we're talking about natural killer cells. This wouldn't necessarily be consistent. Remember, interleukin-10 here is actually associated more with an anti-inflammatory reaction. So this is more used to kind of attenuate the immune response. And remember that we said, interleukin 10, TGF-beta, these are really good cytokines, especially in questions where you have a patient that has like autoimmunity, right? And you need to bring that autoimmune response down. So you want more of an anti-inflammatory effect in those situations. All right, the last one on here, presence of MHC2 receptors. And just recall what these MHC2 receptors are essentially doing. So, antigen-presenting cells, APCs, all right here, are typically the cells that are going to have the MHC2 receptor. MHC2 receptors are not going to be on all of your host cells. They're primarily going to be on your antigen-presenting cells, and they're going to bind to CD4 positive T cells or T helper cells in an effort essentially to destroy usually extracellular pathogens. Okay, so more classic to see, the humoral type response where we're activating a T helper cell, then we're getting, class switching with a B cell to eventually produce a, very specific humoral response. All right, but in general, we don't really think about the presence of an MHC2 receptor necessarily having anything to do with a CD56 positive lymphocyte. If this was a CD4 positive lymphocyte, again, this would probably be more representative answer. Hey everybody, Dr. Viso here. In this video, we're going to talk about B cells. So we're transitioning from T cells to B cells. We'll talk a little bit about just a high-level overview. We don't have to go into as much detail as we went into when we talked about T cell development, but we will build on some of what we talked about with T cell activation when we talk about B cells here. So just to give an overview, again, for step 1 purposes, you don't really have to know quite as much detail as we did when we talked about the T cells kind of going through the thymus, positive selection, negative selection. Not the case really with B cells. Now, you might have learned more in your immuno courses, really for B cells in general in terms of their development. The big thing that you have to know is, again, they originate from the bone marrow, just like a lot of the other hematopoietic stem cells that then go on to form lymphoid progenitor cells. Now, the B cells, like the T cells, will also have precursors. The difference is, remember, the T cell precursors are going to get chemokines that essentially cause them to migrate to the thymus, whereas the B cells, these are going to stay in the bone marrow to mature. That's the key difference. Remember, B for bone marrow, B for B cells, T for thymus, T for T cell. Now, once these B cells mature, and there is a process that goes along with that, but it's not particularly high yield for step 1. Again, you might cover it in your immuno courses, but not particularly high yield for step 1. The big thing here is that once these B cells stay in the bone marrow and they mature, then they become naive mature B cells. Now, remember, you be careful with your terminology here. Remember, naive means that this particular B cell has not come into contact with its antigen. Now, here we're essentially looking at an antibody. And what's really interesting here is that the B cell receptor is going to actually be membrane-bound immunoglobulin. Okay, so in other words, you're going to have membrane-bound antibodies on the surface of the B cell. So you can kind of see that here. You can see these antibodies that are bound on the surface of the B cell. And these antibodies are essentially going to be your B cell receptor. Okay, so this is a little bit different than T cells. T cells didn't have antibodies bound on their surface. And again, we'll talk about antibodies in great detail here, but you can see the antibodies that make up a variable region up here, and there's a constant region on the bottom. And then there's also a couple different mechanical components here that we can see. We can see that there's a light chain here, and then we can also see that there's a heavy chain down here. Okay, well, like I said, we'll talk about all of this in great detail, but the point that I want to convey here is that the antigen binding site, where your antigen is going to bind, is going to be up here in this region. Okay, so the antigen binding site is going to be up here in that variable region, and that variable region is going to be generated through somatic recombination of the VDJ segments as these antibodies are formed. And again, we'll talk about all of that. We touched on it a little bit in the adaptive immunity section, but we'll talk about that in great detail in the antibody video. Now, the other thing that I want you to kind of keep in mind here with B cells is there's usually going to be specific markers that call you to this being a B cell. In this case, CD19, CD20, CD21. Okay, so these are all representative of your B cell. And so when you see these markers, I want you to start to think about B cells. And you might remember, if you watched the video on T cells, that there are specific markers for T cells. And the specific marker for T cells is going to be CD3. Sometimes we'll take, a sample from a patient and we'll run it through flow cytometry just to see how many B cells, how many T cells that patient has. And, just for example here, let's say that I put on this side, let's do CD3, and on this side, let's just say I did CD19, for example. Okay, I'm just using one of these B cell markers. It could be CD20, CD21, right? But the point is that, you have to recognize, okay, this is CD19. So when I see CD19, what are they trying to tell me? They're trying to tell me that this is a B cell. Okay, so when you see CD19, you're thinking B cell. When you see CD3, you should be thinking T cell. And so you can get these graphs where you'll see like a bunch of dots, for example, here, and you might see a bunch of dots over here. And essentially what this is representing when you see these diagrams is it's showing you that, hey, this person has, some amount of B cells and T cells that were identified when flow cytometry was conducted. The point that I just want to hit home here, and we're going to go into this in a lot more detail when we talk about how to read these graphs and that type of thing. But the point here being, if I gave you this and I had a whole bunch of density here at the CD3 line, but I had maybe 1 or 2 dots at CD19, that might suggest that, hey, this person, maybe they have a whole bunch of T cells, but when I look here, I don't really see anything in the area around CD19, right? That might be suggestive of potentially a B cell deficiency, right? Or if it was vice versa, if I had a whole bunch of dots at the CD19 mark, but if I didn't really have any density of CD3, right, then I would be more concerned about a T cell deficiency, right? So that's kind of how you read these. Again, these are just markers to kind of tell you what cells you're dealing with. Remember, CD3 is for T cells, CD19 for B cells. Now, these B cells, not only will they have a B cell receptor on their surface, which we already said is going to be membrane-bound IgM, or in some cases it can be IgG, and we'll talk more about this in the next video. But you can also have the MHC2 receptor. Remember, the MHC2 receptor, the whole point of an MHC2 receptor is so that you can present antigens to T cells primarily. Remember, B cells are also antigen-presenting cells. It's not just dendritic cells and macrophages, right? B cells can also present antigens. So like, let's just say, for example, how would that happen, right? If I had some antigen floating around in the body, right? Here's some antigen that then binds to this antigen binding site on this antibody, right? So it would bind to this variable region. So this is going to be, the perfect match for this immunoglobulin B cell receptor. So then that antigen can get internalized into this B cell. And then what that B cell can do is it can take its MHC2 receptor. Okay, this is my MHC2. Okay, so this B cell also has these MHC2 receptors, so it can internalize that antigen, and it can take that antigen, right, and then it can present it on the MHC2. And then it can take this antigen to a T cell. So once these B cells bind to this T cell, for example, right, then we'd get a proliferation signal, and they would go to the lymphoid follicles, and we talked about this. Primarily go to areas that are very active. Secondary follicles, you'd have germinal centers that are forming, where you'd have a lot of B cells that would be undergoing affinity maturation, isotype switching, and I'll talk about that also in the next video. And finally, activated B cells can become plasma cells. Even activated B cells that don't involve T cell interaction can do this, and they can produce and secrete antibodies, which again, I'll also talk about in the next video. But I think this at least gives you a high-level overview of what we're talking about in terms of B cells. And like I said, next video we'll go into more of the details. Here are the references for this video, and we'll see you in the next one. All right, so we finally have an opportunity to integrate kind of all of the concepts that we've been talking about thus far, and that's when we get into this B cell activation, particularly the T helper dependent B cell activation. But before we do that, let's just take a step back here. So remember, when we're thinking of B cells, we're thinking a lot about humoral immunity. We're kind of looking at the end goal here of essentially generating antibodies and potentially a memory response, right? And so at the end of the day, when I'm thinking humoral immunity, I'm thinking of the end goal here is going to be producing antibodies, right? That's going to be immunoglobulins, and particularly IgG in the setting of some memory response, okay? And the whole point of, immunoglobulins and antibodies, if you think about them, what are they doing? They're floating around usually freely in the bloodstream, right? And they're eventually going to attack or bind to extracellular pathogens. Okay, so that's going to be the key to humoral immunity. We're going to be very effective at targeting extracellular pathogens because we have all these antibodies floating around in the bloodstream. It's not going to be as effective against intracellular pathogens. Intracellular pathogens, I'd want something like cell-mediated immunity, where I can identify viruses or intracellular bacteria using T cells specifically, whereas with humoral immunity in general, this is going to be more effective against extracellular pathogens. Okay, so that's the concept here. So let's just erase this for a second, and let's kind of talk about what we know so far so we can put this all together. So we're going to recap what we talked about in the T cell video, and we're going to go through it a little bit faster because we already talked about it with T cells. But essentially what we're talking about here is, right, we had some bacteria, for example, okay? So we had some bacteria or some pathogen potentially, and then we had a dendritic cell around, okay? So then we had some dendritic cells. So I'm just going to kind of draw it like this. This is my dendritic cell, and this dendritic cell phagocytosed this bacteria, okay? And then essentially presented components of that bacteria to a T cell, right? And so it's going to present these antigens. Usually because it's an antigen presenting cell, it will present them on MHC2. Presents this antigen from the bacteria on MHC2, and it's going to present that to a T cell. Okay, so then let's just say we have some T cell here. Right, so we have some T cell, and so here's my T cell, and this is going to be usually a naive T cell. It hasn't yet kind of ran into its pathogen, and on this T cell is going to have a receptor that's going to bind to that MHC2, and you might know that receptor by now. That receptor is going to be the T cell receptor, and it's going to co-bind with CD4. right? So the T cell receptor, CD4, is going to bind to this MHC2, and this antigen is presented, and then there's going to be a co-stimulatory signal that we talked about in the T cell video, right? There's going to be a co-stimulatory signal, and that's going to be this CD28 binding to B7, okay? And this B7 is also sometimes written as CD8086. Okay, so this is all stuff that we talked about in the T cell video, and it's all recapped right here. We had a bacteria digested by an antigen-presenting cell, presented that on MHC2, usually in a draining lymph node, to a T cell receptor, CD4 positive co-stimulatory signal. Remember your rule of 8s, 4 times 2 is 8. That's how you know this is CD4. And again, it's an antigen-presenting cell, which usually was going to be MHC2. And then you have a co-stimulatory signal that helps this activation of the T cell occur, and that's going to be B7 and CD28. Remember, 7 times 4 is 28, right? So that's how we remember that. So that's essentially the T cell process. Once this happens, now this T cell can say, okay, is this thing you digested more of an intracellular bacteria like tuberculosis, where I'm going to release interleukin 12 and then cause a T helper 1 response, or is this more of an extracellular bacteria that I want to generate more of a T helper 2 response? For example, if this was an extracellular bacteria, which is primarily what we're talking about with humoral immunity, right? That's what we just said. In that situation, we would want to generate a T helper 2 response. So if I want to generate a T helper 2 response from the previous videos, what do I want to release here? Well, remember, T helper 1 response would be interleukin 12. T helper 2 response would be interleukin 4. So this antigen presenting cell might say, hey, we need that humoral immunity, right? Because this is extracellular. I'm going to target this T cell with interleukin 4. Interleukin 4 will cause this T cell to go down the T helper 2 route. Okay, we'll go down the T helper 2 route, and we'll talk about the significance of that in just a second. So that's where we're at. Now, before we go any further, if this is kind of too sloppy for you, I have it all written here too. So you can see the antigen presenting cell presenting MHC2 to T cell receptor CD4, and then you can see CD8086, which is also known as B7, right? So this is also, let me write this in here, this is also B7, binding to CD28, releasing cytokines, right? If it's interleukin 12, then it would stimulate the T helper 1 response. If it was interleukin 4, this would stimulate the T helper 2 response, right? So this is all the things that we just kind of talked about already. And that's where we're at. So we're activating a T cell. Now, the B cell, like I said, can also do the same things as an antigen presenting cell, in the sense that it can also interact with bacteria and then present them on these immunoglobulin receptors. And that's essentially what you see here. So let's just say that we have a B cell here, and maybe there's some extracellular bacteria here. So this B cell might come into contact with this bacteria, or potentially this bacterial antigen, and maybe it's a perfect fit for its B cell receptor up here. Okay, so I'm just going to draw it up here on the B cell receptor. Then what the B cell can do is it can internalize this antigen or bacteria and then represent it on MHC2, just like the antigen presenting cell did. So I'm going to draw an MHC2 here, and so we can put it on, let's just say this is our MHC2 receptor. Okay, so the B cell can place it now on this MHC2 receptor. Remember what we said, what's an MHC2 receptor? Essentially, it's allowing communication with T cells. Okay, so once this B cell binds to that antigen using its antibody, internalizes it, and then represents it on MHC2, now it can go to this step where the B cell can bind using that MHC2 receptor to a T cell. Okay, and that T cell can use its co-stimulatory signal. Again, it's going to be CD4 because we're talking MHC2, and that can help activate this B cell via a T helper dependent response. That's where this T helper dependent comes in. You have a T cell that is interacting with the B cell in order to stimulate the B cell to then go on to have B cell proliferation and eventually the formation of antibody secreting plasma cells. Remember, the end step of humoral immunity is essentially producing antibodies, and this T helper response is assisting with that. It's kind of giving it a huge upgrade in terms of giving the B cell the signals to proliferate, to undergo affinity maturation and isotype switching. Okay, so that's what's happening. This is very similar to what we saw with the antigen presenting cell. It's just when it happens with a B cell, it can then go on to form a humoral response. That's the difference. Okay, so that's essentially what's happening. Now, there's a couple kind of nuances to this that I haven't told you yet. So sometimes when you digest these antigens, these B cells, instead of them directly going to bind to T cells, sometimes these B cells can actually just become plasma cells themselves. Okay, so sometimes this B cell can just become a plasma cell instead of even going to the T cell. Now you might be saying, well, why doesn't it just do that? Why would I go through all of these steps to make a plasma cell when I can just, when this B cell can essentially just do it on its own? What's the point of that? Well, when the B cell becomes a plasma cell strictly because it bound to its antigen and never interacted with a T cell, in those situations, that's a T helper independent response. In other words, the B cell never really interacted with a T cell. It's completely independent. The B cell is on its own. Okay, it didn't need the T helper cell to get activated. Now, the downside to that is typically when this happens, response that's generated is much weaker. Okay, it's much more weakly immunogenic, and that's sometimes how it's said. Now, to organize your thoughts here, you want to think about this in terms of proteins or peptides, right? Proteins, peptides, or you want to think of non-proteins or non-peptides. So things that are like carbohydrates, for example, like lipopolysaccharide, for example, would be a non-peptide. Lipopolysaccharide, LPS, on gram-negative bacteria, it's a polysaccharide. It's a carbohydrate. It's a non-peptide. Things that are non-peptides are not going to be able to be presented to T cells. T cells will only recognize peptide antigens. In other words, they'll only recognize protein-based antigens or peptide-based antigens. Okay, so if something's non-peptide and it's a conserved sequence, right? Something like LPS, something like flagellin. And we already talked about these conserved sequences. We gave them another name before though. Okay, when we first started this video series, we said the name for these are pathogen-associated molecular patterns, right? Or PAMPs. And these are what are recognized by toll-like receptors or pattern recognition receptors, right, to stimulate that initial immune response that goes on to generate interleukin 1, interleukin 6, TNF alpha, right? So these are these same structures. And so you might be saying, okay, well, they set off the immune response. Why do we have to have this T-independent response? The reason is because the T-independent response is what allows us to generate antibodies, like IgM, for example. These plasma cells that are activated independent of T cells because there was a carbohydrate antigen, for example, bound here, they will go on to release IgM, which is kind of like your standard immunoglobulin. Now, it's important to note that IgM is not really involved in the memory response. It's kind of like the initial response to an infection from the immune cells, right? You have your inflammatory response from neutrophils, which is largely driven by pattern recognition receptors and toll-like receptors recognizing LPS and flagellin. The immunological response, right, from B cells is driven largely by this T helper independent response where B cells can bind to LPS, can bind to carbohydrates, right? But instead of going to the T cells, they just become plasma cells and release IgM. What I'm saying here is this is more or less driven by the T helper independent response. That's why it's all in orange here. Whereas if you go up this way, right? If you go up this way, this is more so the T helper dependent response. And a lot of this is driven by the type of antigen. Is it non-peptide like flagellin or LPS? Our pathogen-associated molecular patterns are very classically going to fit into this category, things that are non-peptide based, and not all of them necessarily fit into that category, but that's one way to think about it. Or is it going to be a peptide antigen? If it's a peptide antigen, it's most likely going to go down this route. Now, some peptide antigens will also kind of just generate an IgM response. Okay, but more or less the way that you want to think about it is the way that we have it kind of listed here. Now let's just clean this up for a second. And so with the T helper independent response, right, these specific carbohydrates cause cross-linking of the surface antibodies. That's how this B cell will get activated by non-peptide antigens, and that will lead to production of IgM, which is what we just said. The T helper dependent response involves T cell activation via an antigen presenting cell. That naive B cell is going to bind its antigen to the B cell receptor. That's what we just said here. So antigen binds to the B cell receptor. That's this step. Then the B cell receptor has mediated endocytosis of the antigen. That's this step, right? It internalizes that antigen. Then the next step would be to display that antigen on the MHC2 receptor, which we can see here. So we found the antigen bound to our immunoglobulin, we internalized it, and then we presented an MHC2, and there you can see it right there, bound to MHC2, binding to a CD4 receptor, right? Activated T helper cell binds to that B cell using a T cell receptor and CD4 positive co-receptor. And then the last step here, there's another co-stimulatory signal. And this signal, CD40 ligand co-signal, essentially involves CD40 ligand on the T helper cell and CD40 on the B cell. And this specific step is involved in isotype switching. And that's very high yield to remember that CD40 ligand, CD40 binding, and this is one of them that I would absolutely remember, is involved in isotype switching. So it essentially will cause this antibody to change its heavy chain. Okay, so remember, you have a variable region out here that binds to a very specific antigen, and then you also have another region down here that's the heavy chain. So the heavy chain will tell you, are you dealing with IgE, IgA, IgM, but the actual variable chain is what binds to that specific component of a pathogen. Now, once this B cell goes through this whole process, presents on MHC2 to the T cell, and CD40 ligand interaction occurs, you're going to have specific interleukins that can be released. Okay, and we already talked about this a little bit when we talked about the cytokines. These different interleukins can cause different types of responses. So for example, if this T helper cell releases interleukin 4, that will stimulate mostly IgE, but also some IgG response. Interleukin 5 is going to be IgA. Interleukin 13 is also IgE. So just make sure you're familiar with these, particularly interleukin 4 and interleukin 5, probably being the two big ones I would remember. Once that happens, those B cells can then go on to proliferate. Okay, so the B cells will become activated, they'll proliferate, and then they can eventually become plasma cells, which are essentially going to be antibody factories. And if they, were stimulated with interleukin 4, they might be pumping out a bunch of IgE or a bunch of IgG, for example. Some of these B cells, through this proliferation, will eventually become memory cells, so that if we ever interact with that antigen again, we'll have a much more robust response in the future. Okay, so that's where the memory response comes from. Now note, in the T helper independent response, right, when we went down this road over here towards this IgM, we never generated memory B cells. We only generated plasma cells. Okay, so that's why there's not a really strong memory response when I have T helper independent antigens. Okay, I need T helper dependent antigens to generate a very robust memory response. This is where it all comes from, is from this pathway. Now, the last thing on here is this affinity maturation. So what is that talking about? So affinity maturation is essentially talking about the B cells that have these kind of variable regions up here that bind to specific antigens. What we want to do is we want to make that binding as strong as possible. And so once this interaction occurs with the T cell and the B cell, at some point these B cells will eventually migrate to a lymph node and, like we said, go into the secondary follicles, and you'll have your germinal centers that are going to have proliferating B cells in there. Inside of those proliferating B cells, there's going to be very slight variations that are going to happen to this variable region. And the idea is we know that there's some antigen that is able to bind to that B cell receptor. But what we want to do is we want to maximize that binding. So we kind of know we have the right formula. We just want to make little tweaks to it, and we make these little tweaks to a whole bunch of different B cells. And then the B cells that bind the strongest, right, from those tweaks are going to stick around. And so there's random mutations of the B cell receptors so that only the strongest affinity B cells survive. And again, this is all happening in the lymph node, in those germinal centers that we talked about in the last few videos. And again, here's a lymph node, right? It's going to be more of those pale staining regions in the lymph node where you have the germinal centers. And again, we're just trying to keep the strongest binders in there. And essentially that's what affinity maturation is. Again, we know we have something that binds, we're just tweaking it to get the absolute strongest binding. And again, you get affinity maturation largely driven by T helper dependent responses. So both affinity maturation and isotype switching are a result of a T helper dependent response and not a result of a T helper independent response. That's particularly important. So again, we got isotype switching because of the T helper interaction. We also get affinity maturation because of the T helper cell interaction with the B cell. If I don't have this interaction, if I just go this way, I'm just generating IgM kind of blindly here against that antigen without developing a robust immunogenic response. That's the key, so just keep that in mind. And you might be saying, well, is this applicable at all in real life? I mean, it absolutely is. I mean, if you think about things like vaccines, for example, right? So and we'll talk about this when we talk about vaccines here, but your strep pneumo vaccine, right? There's different variants of it. The PPSV23 variant is going to have a capsular polysaccharide. So the PPSV23 vaccine for strep pneumo is going to have a capsular polysaccharide. And this is what generates the immune response. Okay, so it's going to be the polysaccharide capsule. Remember what we're talking about. It says it right there. Polysaccharide. What's a polysaccharide? It's carbohydrate, right? Go back to your biochem. So this is going to be a carbohydrate. A carbohydrate, is that a peptide? No, it's not a peptide. And if it's not a peptide, then it must be a T helper independent antigen. Okay, so if I give somebody this vaccine, I'm giving them a T helper independent antigen, right? It's an independent antigen. So I know that I'm not going to get this interaction with the T cell. I'm going to go down this road. So do you think I'm going to get a strong immunogenic response if I give somebody a vaccine that has a T helper independent antigen or a polysaccharide? Probably not. I'm not going to get a really robust immune response. That's why we add things like adjuvants to these vaccines, for example, or that's why a peptide vaccine in some situations might be stronger. And in a lot of these situations too, if you're giving these independent antigens, you'll see there's in some situations there might be more boosters involved potentially. But I don't want to get too off track. That's the general concept. And if you got to this point in the video series, kind of building up the material, adding up the layers, this is kind of like the peak. And from this point forward, we can start integrating all of these basic science concepts to understand the pathology and some of the implications. Take 15 seconds to pause the video, read the question, and then we will go through the answer. All right, so we have a 32-year-old male with a past medical history of microscopic polyangiitis and end-stage renal disease. Okay, kind of an interesting start to the question. We're just going to kind of note this here. End-stage renal disease, which means this patient could be on dialysis, for example, presents to the emergency department with a fever of 102.4. Okay, so our patient is febrile here. They have abdominal pain, general malaise over the past 6 hours. The patient states that he is on home peritoneal dialysis. So, there's different forms of dialysis, right? So there's hemodialysis, there's peritoneal dialysis. The fact of the matter is, for step 1 examination, you really don't have to know a lot about this, until maybe you do your first nephrology rotation or something like that. This does make the abdominal pain more interesting to me, the fact that they're on home peritoneal dialysis. So I'm kind of, as I'm reading this, I'm kind of slowly in my mind juggling some of these concepts. The patient recently had his catheter replaced 5 days earlier without complication. Okay, so that's a good sign. So it was recently replaced. Tenderness is noted over the abdomen at the peritoneal catheter insertion site, though. Okay. The patient states that his recently adopted dog playfully bit the insertion site of the dialysis tubing, causing a small puncture in the material just prior to symptom onset. There is something going on here at the peritoneal catheter site, and this dog essentially bit the insertion site of the dialysis tubing, right? So you're kind of thinking in your mind, what are some things that you want to associate potentially with a dog or a cat? Or, one of the things, certainly I can just read ahead here, Pasteurella, right, would be very classic. Rabies is usually not asked about as much in dogs as frequently anymore. Usually if it's like a rabies question, it's usually going to be a different animal, not like the classic cat or dog in most questions, but it could be. It's still possible. It's something you kind of keep in your mind here. If this patient, had salivation and, hallucinating and things of that nature, we might be thinking more along those lines. But we don't really see that here. So we just see tenderness along the insertion site of the catheter. Relevant laboratory studies are shown below. IV antibiotics are initiated and blood cultures later grow Pasteurella. So Pasteurella in general, when you're thinking about this, you think about cat bites, dog bites, not as much scratches. Cat scratches, right? If you're thinking cat scratch disease, you might be thinking more Bartonella. But Pasteurella, usually thinking cat or dog bites. And remember, Pasteurella is a gram-negative bacteria. Because remember, this is how the question starts, and then they usually go on to ask like a specific fact about that bacteria. So you want to make sure you're kind of sharp on the general stuff here. But that's the big stuff. I mean, if you really want to add something to this, you could say, okay, well, it's also catalase positive, right? Maybe if you had a question on, I don't know, chronic granulomatous disease or something along those lines. You just want to kind of be familiar with that too. But again, these are the big things here. Okay, let's look at these lab values. Hemoglobin 11.1. Now, that might seem low, but to me, this is not super alarming. And the reason I say that it's not super alarming is because I know this patient is on dialysis. A lot of times, dialysis patients in general can have a lower hemoglobin, right? Patients on hemodialysis, they're usually aiming for a hemoglobin between like 11 and 12. Now, are you expected to know that for step 1? No, you're not expected to know that. It's just like a little clinical pearl here. This is not a completely alarming hemoglobin. Again, remember erythropoietin, right? Erythropoietin comes from the kidneys, right? EPO comes from the kidneys. And so patients that have end-stage renal disease, right? These patients might be on erythropoietin, for example, or some analog. But again, we're not going to get too off track. Not going to let this bog us down. Again, a lot of these questions have a lot of superfluous information. We want to just get to the point. What are they going to ask us? Just for the sake of this, I'm walking through kind of all of these steps with you. So leukocyte count. For people that aren't super familiar with reference ranges, right? You might look at this and say, oh, it's not too bad. But actually, 3,200 is kind of low, right? Especially like the reference ranges that you're going to see on most tests will be something like 4,500 to 11,000, right? They're usually going to be kind of around here. Now, a lot of people usually when you see a leukocyte count of like 20 20,000 or something, right? You start to think, oh yeah, there's probably an infection or at least some form of, inflammatory response, or there's something going on there, right? Is the patient on steroids? But we got to remember, a low leukocyte count, so like something like we see here, this 3,200, that can also be concerning for an infection, right? It's not just a leukocytosis that's necessarily associated with infections, it's also a leukopenia, right? So you have to also kind of keep that in mind, right? A low leukocyte count is not necessarily normal either. Immunoglobulin M is quite elevated outside of the reference range here. Immunoglobulin A is kind of just on the border of the reference range here, so then yellow. Immunoglobulin G, again, in the reference range, which makes some sense because, again, if we go back to the timeline here, this happened over the past 6 hours, so we might be getting kind of an initial response from the IgM, right? More of the innate response before we develop a memory response. Okay, B cells producing high quantities of immunoglobulin M in this patient are most likely responding to which antigen? Okay, all right. So the answer here is lipopolysaccharides. Let's just talk kind of a little bit about this. First of all, the thing that we have to understand here is we kind of have to go back to the basics, right? So what exactly is happening here, right? So we have some bacteria, right? We have this gram-negative bacteria. Okay, so this is going to be by Pasteurella, right? The Pasteurella is probably coming in through the catheter tubing because the dog, the clue here is that the dog bit the insertion site of the dialysis tubing, and now this patient, since that time, is febrile, has abdominal pain, tenderness, right? We might be concerned even some component of peritonitis. But the point being in this question is they're asking us about the IgM levels. So here's my Pasteurella. So the Pasteurella is in this patient's bloodstream. And so initially, right, the first thing that's going to happen is, remember, because Pasteurella is gram-negative, right, it's going to have LPS, right, in its bacterial wall. And we know that when LPS gets out here in the bloodstream or is identified, this LPS acts as a pathogen-associated molecular pattern, right? We've talked about this a few times. And it can be non-specifically recognized by some of our cells. Now remember, in the B cell lecture, if you've watched that video, right, in the B cell lecture we talk about the B cells, right, this might be a naive B cell that runs into this LPS. And it might identify this LPS as a T helper independent antigen. In other words, this antigen is not a protein antigen, right? It's in this situation, this could be more of a non-specific binding potentially that causes this naive B cell to realize, hey, this is not normal, right? This is going to then cause this B cell to potentially become activated. Now, these B cells, instead of undergoing class switching by binding to a T cell and having CD40 and CD40 ligand come together, this B cell can just go on to become a plasma cell factory, essentially producing IgM. Now, remember, this happens because this is a T helper independent antigen. Let's say, on the other hand, that this Pasteurella infection has some specific antigen on it, right? So Pasteurella maybe has a specific antigen. And now I have a B cell that comes along, right? So this B cell, again, is another naive B cell, but this B cell has a very specific receptor for this antigen. Okay, so the B cell can bind to this antigen. And again, this antigen that we're talking about here is usually going to be a protein antigen. Now, one way that this can happen is this B cell can actually phagocytose Pasteurella to then digest it to get this protein antigen. So that's one way that this can happen, right? This B cell can represent this to a CD4 positive T cell. And we can also get our CD40 ligand binding to occur, right, which would then stimulate class switching of this B cell. So this B cell can then effectively go on to produce, right, specific antibodies, right, after it's class switched. So then it can go on to become a plasma cell. But in this case, the plasma cell, depending on, what cytokines are released and what antibodies we need to respond to this infection, the plasma cell can then go on to produce IgG, IgA, IgE, right, because we had this T helper dependent response. And that was because we had a protein antigen. Okay, now, in general, to generate a T helper response, right, to have isotype switching occurring, somatic hypermutation, all of these things that we talk about, it's difficult for this to happen, in a short time period usually. So usually you're going to get an initial IgM response before that happens, right, especially the first time you're exposed to this pathogen. And that's evident if we look at these labs, right? We can see that there's an isolated immunoglobulin M elevation compared to some of the other immunoglobulins. So it's most likely that it was more of a non-specific reaction with a pathogen-associated molecular pattern than it would be with a specific protein antigen. And for those reasons, it's more unlikely that it would be a peptide in general, right? Because a peptide is a protein antigen. Protein antigens are more likely to have a T helper dependent response, more specific response. Now, I'll talk about these three options in a second, but let's just talk about why it's not lipoteichoic acid. Because again, lipoteichoic acid, much like LPS, is a pathogen-associated molecular pattern. What's the big difference here? Well, remember that lipoteichoic acid, I'll write LTS here, lipoteichoic acid is also a pathogen-associated molecular pattern, but this is specifically in gram-positive bacteria, right? Whereas lipopolysaccharide we know is in gram-negative. So you kind of have to understand here that Pastorella is not a gram-positive bacteria, which is, again, why you still have to know some of the attributes of these different bacteria in these questions, right? Because this is how they integrate this information. Okay, so for that reason, it would be less likely that lipoteichoic acid, right, highly unlikely, that lipoteichoic acid is where we're getting this IgM response from in the setting of a Pastorella infection. Now, going through these other options here, exogenous bacterial peptides. Remember, exogenous bacterial peptides typically are going to come from digested bacteria, much like we just talked about here, right? This B cell can essentially phagocytose this Pastorella bacteria and then present as antigens, right? But going through this process, right, after phagocytosing it and presenting a protein antigen, a peptide, right, we're going to end up with a T helper dependent response. We would expect some elevation in these other immunoglobulins, and we quite frankly don't see that here, right? We have still these immunoglobulins in the normal reference range. Endogenous bacterial peptides, we're usually talking about things like viruses, some fungi, for example, some intracellular bacteria. We really don't see anything here suggestive of that again, and the isolated IgM elevation, again, would be more suggestive of a T helper independent response versus a T helper dependent response. Finally here, host peptides. Sometimes you can see this, if a patient has like an autoimmune reaction or something like that, they might start presenting their own host tissue to their lymphocytes. This is basically why we go through this kind of arduous process in the thymus, right, of selection in general to avoid things like this happening. But again, this isn't particularly relevant to the fact that this patient appears to have an infection of sorts that isn't necessarily consistent with an autoimmune reaction or a peptide antigen in general. So lipopolysaccharide here is the best answer. Hey everybody, Dr. Aviso here. In this video, we're going to talk about antibodies. And I feel like this is a pretty good graduation from the B cell lecture where we talked about plasma cells that are going to go on to produce these